Hello students, welcome to Sarasa classes. Today our topic is cell organelles and I'm going to discuss about some important cell organelles in this video. And at first I'm going to discuss about endoplasmic reticulum, that is the filamental structure. And I'm going to discuss this through some points. So now let's get started. The first one, the first point is Garnier for the first time had described them as filamentous structure. Has described them as filamentous structure. Ergastoplasm, which is known as ergastoplasm. Now, the next point is, so it was first described by Garnier and was known as ergastoplasm. Now, they were first discovered by Claude. They were first discovered by Claude in 1951 under electron microscope. Under electron microscope. Now, the next one is the term endoplasmic reticulum was first. The term endoplasmic reticulum was first coined by Potter L. in 1945. Again, endoplasmic reticulum, that we can write it as ER, is the abundant in liver, and other actively synthesizing cells and other actively synthesizing cells. Now, endoplasmic reticulum, or ER, is absent in RBC. Endoplasmic reticulum is absent in RBC over embryonic cells and prokaryotes So we can't find endoplasmic reticulum in RBC, prokaryotic cells or prokaryotes and embryonic cells. Now the next one is, there are two types of endoplasmic reticulum. There are two types of endoplasmic reticulum. Rough endoplasmic reticulum and smooth endoplasmic reticulum. That is rough endoplasmic reticulum and small endoplasmic reticulum. Now again it can divide it into two parts, tubular forms with ribosomes, tubular forms without ribosomes, and the next one is, it is more developed in cell actively engaged in protein synthesis. So it is more developed in cells actively engaged in protein synthesis. Now it is abundant in the cells engaged in glycogen and lipid metabolism. So it is abundant in the cells engaged in glycogen and metabolism metabolism. Now again, I'm going to give some features of endoplasmic reticulum. So here are some features of ER. Now the first one is ER or endoplasmic reticulum. It occurs in three forms, such as tubules, next is vesicles, and another one is cisternae. Now next one is endoplasmic reticulum originates from the ground substance, that is haloplasm or enfoldings of plasma membrane or enfoldings of plasma membrane or evaginations of nuclear membrane However, exact nature of origin is unknown. Exact nature of origin is unknown. Next point is endoplasmic reticulum that is present in retinal cells is called myeloid bodies. Next point is microsomes are the fragments of endoplasmic reticulum. Microsomes are the fragments of ER or endoplasmic reticulum containing ribosomes. containing ribosomes. Now, those present in muscle cells, those present in muscle cells are called sacroplasmic reticulum, are called sacroplasmic reticulum. Now, the next one is they contain large amount of RNA and phospholipids. They contain large amount of RNA and phospholipids. RNA and phospholipids. Now, I have discussed what is endoplasmic reticulum and what are the features of endoplasmic reticulum here. Now, I'm going to discuss about another important cell organelle in this video, that is known as Golgi complex structure. That is known as Golgi complex structure. Now, first I'm going to discuss about the origin, or what is actually the Golgi complex structure, and then I'll discuss the characteristics. So, an Italian physician Camillo Golgi in 1898 first recognized this structure in nerve cells. This structure in nerve cells. Now, this cells was of burn out and hence named after the discoverer, that is Golgi, after the discoverer. Now, these are also known as lipochondrion. These are also known as These are also known as lipochondrion. Golgi body or Golgi apparatus. Golgi apparatus. Now, I'm going to discuss about the features. Now, it is found in all the eukaryotic cells. It is found in all eukaryotic cells except red blood cells. And seed tubes of angiosperms. So we can find this Golgi complex in all eukaryotes, but we cannot find it in red blood cells and sheath tubes of angiosperms. Now the next one is, they are very well formed. They are very well formed and larger in nerve cells and gland cells Next characteristics is, it is also absent in prokaryotes. It is also absent in prokaryotes. Certain fungi and sperm cells. The next point is, the Golgi complex is morphologically very similar in both plant and animal cells. Morphologically very similar in plant and animal cells. In plant cells and in invertebrate and in invertebrate tissues dictyosomes are disappeared throughout the cytoplasm. Throughout the cytoplasm. So here I have discussed some features and the sources of the Golgi complex structure, and I have also discussed about the endoplasmic reticulum here, the features and the origins of these cell elements. Thank you.